I'm not gonna.
Your turtles seem like they love it, man. Those things are going nuts.
Yeah, the turtles love everything. Seems to be thriving, man. You know, the lactic acid bacteria that I've been using that in the water, and that's made a huge difference. You know, I wish I had known about that when I had aquariums.
Yep.
Because that would have solved a lot of my problems in the aquarium world.
See my camera still, right? You see this?
Wow, look at
Okay,
that.
which is we clear right away. Everything settles. You got a little sand down there. Your silt is there. You got your organic matter floating up top. Keep in mind that the darker this is, the more humic substance we'll see in the sample. It's a one. What's
If
that?
it's silty though, you'll have that blackness for a bit, and then it just all settles down.
Yeah, he's silty as hell. That's
Yeah.
exactly right. So there, you can see. I don't know if you can see the sand there,
Mm-hmm.
but yeah, digging it. And we jump it off with a nematode. So
Yeah.
there's. I just turned it on. So
Of course we do.
This is his though. So I'm at 40. I'm gonna just jump in on this nematode real quick.
Yeah.
Mo is the nematode whisperer. No matter what sample he pulls up, it could be like something dead from the moon. There will be nematodes in it.
Leighton said he saw 6 different types off one slide.
That's interesting.
And I'm glad to see this one as soon as you pulled it up. Like I was like, okay, it's cool. So
this one I'm at 200 now. Its behavior is bacterial. The inside of it may look bacterial. You see the little dots.
Yeah.
His face is kind of buried down, so we're not getting a clear shot of his mouth.
Yeah.
He's grazing on bacteria. You see all the dots around him?
Mm-hmm.
That's all bacteria. I don't know how well you can see that. I'm at 400 now.
So the way he's twitching around too, his face looks like he's got some pretty fancy lips hanging out there, but he keeps facing away from us.
Yeah, so he's going. We got like 15 I think it's micrometer or nanometers between the slides. So he's actually has a little depth and that's why his face is digging deep and up and down. But he's grazing on bacteria. That's the behaviors of them and the size of them. So we can I'm gonna pull back on this guy right now. Sample 1 you said is
that was right when we walked in, like right when you walked into my room. That's like the first that's the first pocket. Like I call that pocket 1, and then we come around the sides, pocket 2, pocket 3, and then in the center is pocket 4.
So I'm gonna mow this real quick. Mow mean I'm at 4x magnification. So if you had you're able to zoom in 40 times, there's another nematode. This is why I do that, right?
Yeah, it's easier to pick them up.
Right, at 40 it's easier to pick them up, and then I'll dive in on the nematode. For nematode hunting, that's where we want to be.
Yeah. I'd say.
So are you seeing anything else that's other than the nematode, like ciliates,
bacteria?
Right now,
any fungal?
Right now, bacteria. I haven't seen hyphae yet. Again, we just turned it on. A lot of small aggregate, which is going to be there's 2 of them.
So can I ask a question?
Ask away, bro.
So we're looking at the first layer. So I noticed that there are things behind what we're looking at.
Yeah. So if I go deeper, for example, we're going to see more bacteria.
So how many layers are there? Like how deep can you go? Like is 400 like?
Yeah. So at 400, where the deeper the layers, you'll find junk that settles up. It seems that usually there's a sweet zone. Like, see here, I'm going back up now.
Yeah, that's what I'm saying. Like, there's a middle zone. Is there a hot spot in there?
Yeah, this is your hot spot here, right?
Okay.
Yeah.
And so from here, we can tell if there's something else deeper. You can see the shadows in the background, and they're not shadows, but they look like shadows.
Yeah.
And as I zoom in there, like I'm looking at this right here as a reference, right? This little piece here. If I pull back out, you see that's a shadow back down there.
Yep.
But now we're going a little deeper, right? And now we start losing the view of the nematodes and we start getting into that depth.
Gotcha.
And then as I pull in deeper, you see more bacteria and everything starts pulling away.
So all those black dots, that's bacteria.
For the most part. I mean, we can say it's bacteria, particles.
Yeah, some little micro particles, yeah.
But yeah, I definitely got a good consistent so far. Again, we just put it on here. So I'm looking at where is my at over here?
I think it's pretty viscous as well, Mo.
We're at 3 nematodes already. You
I'm
haven't even moved anywhere.
That right there.
Yeah.
Whatever that is. And I'm going to take it to 400 real quick and see if that might be.
And this is a soil sample and it's from Yellow Skunk Farms?
Yes.
Yeah. This is soil sample number 1.
That to me looks biological right there, bro.
That rod right there, or the circle right there.
Right here. Can you see my mouse? Yeah, right?
Yeah.
So that right there is
biological too, Mo.
Yeah. What it is, I don't know. That's in that shape usually. And then this nematode looks like
is it protozoa of some sort?
I don't know if he's a bacteria feeder, bro. I think he might. I don't know.
That's pretty defined.
He's full. I know he's full.
I don't see a basal ball, but I definitely see a very well-defined throat going down.
He got a full belly.
Yeah, that's bacteria. Yeah, he's definitely full. That's probably why he's not
moving much.
He's one of those fat chilled out. He's a slow wiggler.
So I'm gonna just look.
It was
right here, bro.
Yeah, there was the test date you just passed up there.
I did.
Right there.
Yeah, there,
oh, heck yeah. There you go.
Oh, that thing's cool as shit, dude. I gotta get me a microscope.
Testate amoeba.
Yes, you do.
Testate amoeba. Do you know you need some information on these guys or what they do? They build their own shells. They're the I don't know, the pinnacle of aerobic microbes that everybody's always looking for. But
so that means that there's not too much water, but there's just enough water. Is that what that's telling me? When I see guys like that, that's telling me that the system that right in that area is like perfect amount of water, moisture.
You're in you got aerobic conditions. It's not anaerobic, but we could come through this. We could find some facultative anaerobes coming through here at some point. But yeah, this is a good sign of aerobic soils, and you can kind of see he's pulled some organic matter back into the front of the shell to kind of protect himself, and since he just got shaken around like a maraca.
Your microbes are just chilling. That's what happens.
Yeah, that's another great thing. Very calm sample.
Can't cannabis grow.
Well, the plants look super healthy, and I haven't seen any, pest or any type of damage or anything like that, and I'm just like, I haven't done anything, and I'm just like, the soil must be just on fire. And what I'm seeing on the edges of the creek, every day it's just changing. It's constantly turning, and there's so much life. I mean, I can just move just a fingertip of soil and a worm will be there, So I know if I see it's just nothing but worm castings and just life, like you said, springtails, rove beetles. I mean, the whole 9 yards is there.
But as far as closed systems go, like I seen your YouTube stuff, you got good stuff. Like you got a whole ecosystem going on.
Yeah. That's I wanted to create a home for these guys. So if
absolutely,
we gotta build a home, and they gotta have a place to chill and hang out and smoke and do what they gotta do.
Bro, is that a piece of something, man?
I say it almost looks like another a very odd testate amoeba that's pulled organic matter in an opening right there. It's not because of the way it fluoresces and everything, but
I've never seen a test.
Yeah, what's going on at the top there?
It looks like it's been ripped off or something. Yeah.
Yeah.
Is it like a partially decomposed like plant material?
I expect to see.
I don't think. I mean, it looks like it's got bacteria and other stuff. I have no idea. It's like it looks like it was alive at some point.
Yeah.
So Josh, this here,
trichome.
Josh, these things here.
Yeah.
That's yeast, right?
Oh, other Josh.
That's your yeast. These little things. Yeah. Sorry, there's a couple of Joshes here, right? Yeah, that egg shape there. That's going to be your yeast.
Okay.
And then keep in mind that when you see dark, let's pull it back a little bit. You've got 3 major colors here, and this is probably the best example of it. Dark black, dark brown, and light brown.
Yeah.
The black is going to be your humic. The brown is going to be your
Well, the dark brown.
I'm sorry, the black is going to be your carbon. The
Yeah.
dark brown is going to be your humic. The light brown is going to be your fulvic, right?
And is that testate right there, Mo? Up against that piece of organic matter?
Yeah, it's definitely a testate there.
Or at least a testate, maybe not, or at least the test portion of it.
It was a test, right? It could be that it's still in there. It looks like something was in there.
What's that purplishy green, like fluorescent color there?
That's the
probably
a mineral, maybe.
Well, but it could be also the light, the light, the way the light's
reflecting off a piece of sand or something. Okay.
Yeah.
Felt it was pretty small.
And what's that rod thing? Is that another?
That looks like a
nematode.
What is that one point? It looks like it's cut in half.
It could have been.
Looks like it's a cut.
Yeah.
You're also aquatic, bro. So that changes the game here. Like my
my
eyes right now are prepared for that because we're gonna see things that we're just like, what is that? It's endless in aquatic systems. I observe a lot of that. But take note of the color so far of your organic matter. You got a lot of light brown. Means your pH is probably, if I'm looking at this and I'm going to take a shot at your pH, you're like at 6.6, 6.8.
That's where I want to be.
And I'll have a meter. I will do all that since I have your samples here. So I'm going to hit it with epifluorescent light. Yeah. And this is going to show nutrients that are attached to the carbon, right?
Oh, what the fuck is that?
This is a piece of organic matter with just nutrients attached to it. You know, this is what I'm saying. This is where your roots and your microbes come and they start. When your plant says, I need fucking calcium, it's going to come to one of these pieces of organic matter and start mining it.
Yeah.
And if you understand how that works, then great. But if you don't, I have a very simple way to explain this shit the way my brain accepts it. And that bacteria is going to go up to a piece of organic matter and it's going to send out a bunch of little enzymes that it called knives. And those knives are going to start fucking hacking away at organic matter and then start absorbing what it's releasing. There's another piece of it.
Kind of like me eating a piece of steak.
Exactly. No
like Edward Scissorhands?
Yeah.
Your knife and your fork are your enzymes. Right?
Cleaving nutrients.
There was a testate amoeba that got crushed. He could have gotten crushed when I shook up the sample.
Rest in peace to the testate.
And everything spewing out of that is but look at how peaceful everything is. It's in harmony, bro. You get it? That's a big deal. And then to be at this level of biology already with that harmony, it's like a settled worm casting is very peaceful like this.
So what does that mean when you say like does that mean that everything is broken down and just waiting for the root to come or waiting for microbes to come accept it?
It's kind of balance where it's got the biology, everything is doing what it needs to do. Nothing's panicking, trying to get everything fixed all of a sudden. Like it's just it's not in a crisis mode anymore. Right.
And do you think that that's because of the lactic acid bacteria that
it's a mixture of everything, your entire system?
The whole thing, right? You got
it's just everything comes together.
Soil, good nutrition, probably.
Yeah, he's got cover crop. I mean, he's got cover crops in there.
Yeah,
he's got roots in the ground.
Yeah,
but I want to note too that like this is the difference between getting somebody to count your biology or a machine to count your biology. And looking at your sample is that we see the chill, we see the diversity, we see the interactions, and you don't get that perspective with numbers, right? And this is where, and to me, this is the more important part of getting the feel, right, and the interpretation from a human perspective of regeneratives. And that's compared to just getting, hey, I've got a fungal bacteria of this. Like, okay, great, but is it happy?
Right. I think those are organic matter or hyphae back there we're seeing.
This is a piece of hyphae.
That's what I thought too. I thought we saw another one.
At this point, this is what I'm looking for. Because if I were to wrap up this evaluation, I wouldn't be happy without finding more of this, right? We want to be fungal. So
which one are you looking at now? That long
brown one right in the middle.
Little brown strand right there.
Okay.
That is a great example of what we're looking for, just extended, longer, more of them. You see the septated piece going across here, the little black line.
Yeah.
The color is solid. The size is big. That's when you're fungal dominant. You're rocking and rolling, which is a great thing because that just means there's room for improvement, dude. This is insane. Imagine your bed just on all cylinders, man. That'd be crazy.
So
how many?
Sorry, go ahead.
Sorry, I was just gonna say. Sorry, hi guys. I'm new here. This is my first meeting. I'm Adam. You guys all know me.
Adam.
I was just wondering really quick, Maurice. Yeah, what's up, man? What's going on, bro?
Bro, been a minute. What's up?
Yeah, I'm glad to be
Hey,
here,
Adam,
man. I'm
I've
loving
seen
your.
you before. Have you talked to Mark who was growing?
He's out there with Leighton
Yeah,
Morrison and all that,
Leighton?
bro.
Yeah. I think
nice.
okay.
Yeah,
That's okay. Awesome. Good to meet you, man.
yeah. Heck yeah, man. I'm good. So we're looking at a sample from you, right, Josh?
Yes.
And okay, how many drops did you use? Was it like 1 or 2 drops of this sample?
Yeah. So I cheat, bro. I don't cheat. Look, I got this. I can pull
Okay.
4 drops so that I'm not horsing around with one little drop, and then I got to worry
Okay.
about it drying. I like to take my time. I like to stretch the mic, bro, and
Okay.
otherwise we start drying, it starts collapsing, and we got to go run around and pull another.
Right. I was just saying that just to see like on the whole Elaine Ingram like perspective with like how many nematodes we saw, because we're already seeing quite a bit and stuff too. So then we could put that in the essence of how many drops you have on there, and
Well,
then
now
kind of figure out, right? Or
in respect to that, dude, I hadn't even gone through like 2 fields of view.
right.
3 fields of
Right.
view, and we already see the gang of nematodes.
Is that a dower? Is that a dower right there? Oh,
That's a cyst here. Whatever.
no, down below that. Is that a dower laying there in the background?
I don't.
It's kind of fuzzy just below that cyst.
Well, if it is, it's gonna fucking jump here.
Wow.
That's not a
Dark
dower.
light.
It's organic matter right there.
That's either organic matter or microplastic, one of the two. And I would call that organic matter.
Gotcha.
And so we can walk Josh through with being able to actually like circle around things we're talking about, because we're used to seeing all this stuff, but he's
What
not.
is that?
Yeah, no, I've never seen
Is that an Arcellamo?
I don't know. That's definitely not Arcella. I mean,
I've been watching.
Yeah, it's got a really nice
those videos, and I know kind of, but this is the first time I've seen my stuff.
Yeah, that's what makes it great.
But no,
it does have that like long-standing kind of quietness to it.
Yeah, he's in harmony,
like mentioned.
Yes.
That only comes with time. Like you can't game that. You just, and most preps are not going to look that way. Like if you're making something that you then put into your soil, it's going to be higher density and like have a specific activity direction because it's dealing with excess nutrient, as it were. But once you're into mature soil, it's not about having excesses. It's about that mediated force, like longevity. So yeah, you definitely have that, especially on like the bacterial and the yeast end. Like I'm sure a lot of those smaller particles are yeast. Like Mo said, getting some more fungal hyphal, like because the bacterial and the yeast layer is like the substructure, and then the superstructure is like those fungal webbings, and those are just different inoculations into like the crops that are within a system and stuff to move towards
that fungal portion of it, for what he's doing, he's created a tropical environment completely manually, which is amazing. You've accomplished. I mean, this is amazing.
Yeah, that's right. Because
that's a test
I was going to say it's
but I do want to. I mean, just
cool
I haven't seen your whole setup.
Bro, I have no idea what that is.
But
that is bugging me out.
Like fungal, like overly fungal, may not be your target. Like you may want more fungal, but when you get spores and hyphae, like. But your environment may not necessarily be. You aren't going for old growth forest here.
Okay.
I can't tell if this test state is spewing out.
Yeah, and when you have a tropical environment, you're not going to get the cycling that causes those hyphal. So then, really, your fungal is coming a little like more from yeast and like those healthy tropical systems.
It doesn't.
The
goal is not old growth forest.
I can't tell
really
if
do
this test
have
state
the
is
nice, quiet, long, steady.
These test states usually blob out like that. You see this blob? That's what they look like inside, right? So sometimes they'll come out and you'll start seeing pseudopods. I can't tell there. Even when they die, they'll do that. There's many different ways I've seen that. But that's definitely a cool little test state amoeba.
Yeah.
I like that thing. I want that. You see this here? This is bacteria that's glued itself to this piece of organic matter, right? So this is when it starts gluing itself to things. This is when it starts creating soil, really aggregates of soil. If you were to start seeing this everywhere, and I'm going to use Matt Power's word for it because I think it's the perfect word, it'll start looking feverish, like something's wrong. When bacteria starts quorum sensing like that and communicating with each other, and they need to start wrapping things around, they'll do that if they run into a piece of aluminum, for example.
Okay.
They'll come together, dude, and they'll communicate. They'll be like, we need to lock this thing up. And they will wrap themselves around it and basically neutralize it. So if you start seeing a bunch of this stuff, I start worrying about it just because I've been taught by Matt that, and it makes sense after all the samples that I've seen.
There's probably like a heavy metal in there. It's trying.
No, it could be that they're just handling.
No, he's saying within a broader context, it can still be concerning, but that behavior mimics itself. It's like it's good when there's a herd antelope, but it's bad when there's
an insect part right there.
So,
what is that,
bro? That is.
Uh-oh.
Is that a twig?
That was a big ass.
No,
it's a leg.
It's a spider hair.
Oh, yeah.
Is that a
leg?
That thing was alive, bro. That's a leg or something. That's a big ass leg.
This has so how does the recirculating water interact with the soil in the system?
200, okay.
Yeah.
Oh, you got to see the videos, Landon. It's pretty awesome. He's got like a little lagoon and running stream in his grow room.
Yeah, I've seen some of the video. I just wonder about like how much is it directly interfacing with the soil? Like, is it like a riverbank all the time, or
only in certain places? So if I try to do like that was the problem, like regulating the water. You know, how could I regulate it and keep it, you know, maintain the consistency amount of moisture that I needed into the soil beds. And it came down to creating a bridge with that sand, silt, and clay and organic matter. And it was all about removing that organic matter because that organic matter was the bridge, so to speak. And the more organic matter I added to the side of the creek, the more wicking action I got up into the soil pockets. So I just created a bridge, so to speak. So wherever I added that organic matter is where I then was where it would come up into the soil bed. But then once it came into the soil bed, to spread the water out evenly over top of the soil pocket, I just spread the same amount of sand, silt, and clay that was in the creek and just spread an inch layer of it. Instead of mixing it into the soil, I just laid a layer of it over top of the soil bed of the soil itself, and then put organic matter on top of that. So the water that's being wicked up out of the creek by the sand, silt, clay, and organic matter, and then it enters into the soil bed, and then it wicks out across, spreads out across the top of the bed. Now, this changes constantly because the worms have now completely turned that inch layer into a layer of worm castings. So after about each run or so, maybe 2 runs now, I'm thinking I'm going 2 runs now, where I have to add another layer of sand, silt, and clay coming up out of the creek to get that same wicking action that I was getting. But once it started, it's like nonstop. I mean, it was like way too much at first.
Yeah, that makes sense.
And then once I got roots in the system, once everything started to dial itself in, it was really about getting the plants in there. Once I got the plants rooted and going, they started growing. Then they started pulling water, you know, and then so basically all that excess water I was getting is just being absorbed by all the roots. So there was a time frame of a couple of months in there where it was hell. But now that I've got the roots, I've got the grass, I've got the algae, I've got all these things.
There's your dower.
And during what you called hell, that's where you would see more of what Maurice was saying about the fever pitch. Because
I'll show you hell, bro. I'll show you chaos.
Heavily reduced conditions, like waterlogged conditions, and that's where they get in that fever pitch because there's excess energy.
Yes.
And the plants are actually like mediating and tapering that down, and that's what you're seeing with like the slow roll of that substructure that we're looking at of bacteria and yeast.
Bro, that's a dope ass test.
Like they found their even stasis, or it's not a stasis, it's equilibrium. But
exactly. I mean,
but guys,
really
I don't
cool.
And the plants show it. The plants really do show it. Like I could see the difference. Like this run, this is the 4th run versus the 1st run and 2nd run. I can see a huge difference. Like the pest pressure,
and the sample does look very aquatic.
Hey, real quick before I move on, that is an encysted dormant nematode. At this, this here I believe is a mycorrhizal fungi spore. At least
Spore,
that
yeah.
is what fits in that
Yeah.
explanation of it.
That brown thing in the center there?
That brown thing, yeah.
Yes.
It's a larger spore. It's round.
Single spore.
Yeah, that's what an indication that it could possibly be a mycorrhizal. But there are so many spores out there. I just point that out. Do you inoculate with mycorrhizal fungi?
No.
Good. Probably got that already from your pond or your river.
Mm-hmm.
This here, potential spore.
Mm-hmm.
What's the little spinner there?
He's
He's spinning,
let's go deep.
Oh.
Let's go deep.
Yeah.
He's barreling.
He's a dude. He's doing stuff.
He's barreling.
Barreling. I can't make that out.
To speak to the teaming thing, it's not that in every context everywhere that's a bad thing. It just does speak to high energy, high anaerobic. So like if you're doing a biofertilizer, that might be exactly what you're going for because you're trying to like reinvigorate a low energy system. But when we're looking at a soil sample that we're trying to grow plants in long term, we want to see those signs of maturity, like we're seeing in this context. This is a little more waterlogged than what you would see in most soil systems across landscapes. But this is kind of what you would hopefully see near like creeks and waterways and stuff. Is this kind of range. So there's always going to be those variations in context, but if you were seeing that teaming in a mature soil space, that would indicate like an anaerobic pocket, which might be concerning for your plants.
I'm betting you'll see more anaerobic stuff in the fourth sample, like in the center bed, because the center bed is where, Leighton pointed me towards, he was seeing things towards the more waterlogged side, and he was like, you need to kind of reduce the water flow in there. And sure enough, I went in and I poked around and went around the whole creek. The center is connected, all the way around pretty much. So where all the other soil pockets are just on one side, the center is surrounded by it. Correct. So I had that sand and silt coming up over the creek and into that pocket too much. So basically I just had the valve open too far, and I needed to remove some of that sand and silt from the edge of the creek, disconnecting the bridge from the soil, and that reduced the water flow coming into the soil pocket. So I just did that a week ago. So we're going to see, I know it's going to reduce the water. I mean, that was because when I stuck my hand down there, I started digging around low. I was like, holy shit, yeah, it's a lot of water. And it was crazy how Leighton could tell me that. He's in California, never seen it before. And he looked at a piece like what he's doing right here, like what we're doing right here. And he was like, yeah, you got too much water in there. Like, and I was just like, how the fuck do you know that? Like how, I mean, that's insane to me how you can get that information just by looking at a slide here. And I was like, that's insane how you could tell me that. And then I go investigate, and sure enough, there's too much water going in there. And I was like, okay, he's right.
Bro, Leighton's a monster, bro. He's been doing this shit forever.
I was just like, well, I mean, that sold me right there. I was just like, holy cow, man. I was like, you can tell that from just looking at a little tiny piece of.
Well, you get it with compost too, because like if someone like made a compost and kept it dry the whole time and then wet it 10 minutes before he handed it to you, you're gonna see that. And if someone kept a compost really, really, really nice, and then it kind of dried out to like 40% on its way to you, and you get it and you're like, dang, this is kind of dry, and you re-wet it and it bounces back.
Yeah, that makes
super vigorous. You're like, oh no, that was just like right at the end. You let it slip because shipping or whatever.
Right. So, Josh, before I jump to the next one, man, this is a polarized film that exposes minerals. I can't tell you what they are just because that'd be like a $40,000 microscope, but definitely shows off.
This shows, so what's glowing is
everything's glowing, and every different color is a different type of mineral.
Okay. So this is a heavy, is this what this would be considered heavy in mineral or high in mineral?
It's balanced, bro. This whole thing just looks balanced, dude. Honestly,
it's balanced. Okay.
Which makes sense. I mean, the whole sample's balanced, and then look at your system. You've got a living ecosystem there, dude. That's just like doesn't surprise me one bit.
Do you have your polarizing film set right now for complete extinction, or are you in an angle of that?
I got two pieces of film, Henry. Nothing technical that I can even dial it in.
Well,
It's
no. So if you take the two pieces of film and you turn them at a right angle, you should get extinction where it turns black and you can't see through it. Is that where you're at right now? Or are you at an angle of that?
Well, okay, so I just slapped them back on, right? And I'm twisting, twisting. There's like a small window here that, bam, here's where I start fading out.
Yeah, where it's black.
Makes me want to look at some creek samples.
That's extinction.
Okay, that's called extinction. Awesome. I didn't know you were here. I'm glad to see you here, Henry.
Yeah, it's nice microscopy. I had a question about the sample though. So how close is this taken to the actual stream? Where did you pull the sample from?
That's a great question, because that's exactly where I was kind of getting ready to go with this, because the closer I get to the creek, like it comes over like the creek wall, and then it has to come up over the creek wall and then into the soil bed. So that's how I separate the soil from the creek. So the soil is touching basically all that worm casting there that's on the edge of the creek. And then, so I did 4 fingers basically. So I just put 4 fingers down from the edge of the wall, kind of almost right where the cannabis plants are planted. And I dug down a little bit right there. So it's moist anywhere I dig in the bed. Like I can come all the way to the rock wall, like as far away from the creek as I can, all the way to the rock wall, and dig down. It may be dry about an inch, maybe 2 inches, and then the deeper I go, the wetter it gets. So that's probably from the initial, when I first set up, that initial inch of that sand, silt, and clay that I laid down on top of that soil.
You want to hop to 4?
Hey, Mo, can you go up a little bit? I can't tell if that's a potential long piece of actino right there. If it's just something on
that's a slide, man. That's the edge of the slide.
Making sure, because I know I saw something earlier that looked like a piece of actino, and I've seen like 4, maybe 5 pieces of fungal hyphae.
Let's run 4 to contrast here. That'd be cool.
Yeah, 4 is the center, and 4.
There were some really good fungal strands in there.
That's going to be the driest soil too, ain't it?
Pretty good color too on
fungal. Well, through the bag. So 4 would be the driest, or 1 would be the driest?
I think 4 should be the driest one.
Okay.
Which one is this that we're looking at?
Bro, this is number 2, dude. This is like action packed.
Yeah.
Whoa, what's this? Holy crap.
Yeah, look at all the spores right there.
I think what Richard
20
was ready to point out with the ammonium idea was the zeolite.
There you go, zeolite, yeah.
Okay, it's zeolite, yeah.
Zeolite. Yeah.
So
I had those 2 backwards, Jeremy. Thanks for catching that.
If you want to buffer ammonium in a aquaponic system, zeolite has an affinity, or a cationic ionic bonding affinity. So every particle through the soil, water, everywhere has a different profile or a preference in a hierarchical order of different elements. I might choose different elements than you might. Same thing. So everything has a different CEC priority. When we look at the word CEC or phrase in the soil, it's a very generalized term of the total cation exchange capacity.
That's like lignin, bro, or what is that being broken down?
Yeah, that's like cellulose or something.
Cellulose. But you've got
you got a testate amoeba here. You've got a testate amoeba here. You've got a big ass spore here. You've got a cyst here. Another testate amoeba here. You've got a fungal spore.
You might actually that might be your basidiomycota right there, Mo. That almost looks like a bracket connection right there.
Yeah, that one does look more basidia with the
bottom one.
Branch and
that bottom one right there. If you can zoom in on that, that looks like a clamp connection on a
pretty well septated. Yeah.
Yeah, and it's a right color.
So is this a good sample?
Oh, this is a gangster sample.
Yeah, this one's like, again, dense and mature. Not like that dry one.
This is the heart of your
not like that other
so
crap.
far. Like, this is where all that.
Where's this one? What sample is this, Mo?
Sample number 2.
So this is the second. This is the corner. So when this is around the corner. So this is where this is the bush. This is where the bush is.
Here are your yeast. You say lactic acid bacteria. The yeast from that There's a
What's your
fungal
dilution,
fungal
Mo?
hyphae there. Like 1 to 7 here, which is insane.
Okay, yeah.
1 to
I
8.
mean, this is really
It's
good.
fucking rocking.
Yeah, that's the
It's
third
really
different
dense.
species of fungi that I've seen in this one sample.
Yeah,
This is what I'm talking about. Is you can have like these are the fungal counts that I was talking about in the
wing right
first
there.
one that we looked at.
That's probably
Where you want to have this more of like lift of this fungal presence, and the fungal I think is going to be able to pull through more when it has that sweet spot of wet but not sodden, because when it's over waterlogged, it's going to push it out. It just can't breathe in
How do
those
you test
conditions.
your soil for its moisture saturation point?
I don't.
So,
I don't
have you
test it
seen
at all.
Dr. Elaine? She describes the simple method of you just using your hand to grab
that's
the
basically what
soil
I
and
do.
squeeze
Yeah.
it
Yeah.
till you get a drop. That's actually a very scientifically valid method.
Cool.
So
there too.
I always tell people to use that as a benchmark to gauge.
Those are some interesting conidiospores. Yeah, you have a lot more spore count and hyphal action here. So the fungus is just all the fungal players are able to cycle more and more, and you'll notice that your amoeba density is also higher.
And these little guys too. These little guys are just
when you have more fungal content, you have more total food, like for the actual volume of space. They're the ones who actually make more food total happen. So that's why you're seeing an even more robust predator count, because they have the energy there for it. And that's why that's going to correlate to healthier plants.
Shit.
Yeah, the diatom is cool shit.
That thing blue.
The diatom is so cool.
Red right there.
Shows that you're in the aquatic, like
that DE you were talking about, the diatomaceous earth. Those little things right there are what it's made up of. That's a diatom.
Okay, gotcha.
They're actually like really sharp and razor-like.
Yeah, I was gonna say they're like razor blades, right?
But that's only when they're dry and not occupied by some form of water that then relieves that pressure. When they get up against it, when it's wet, it's kind of lubricated. It doesn't cause the damage.
But then look at the size of the aggregate and everything. Did you find that in castings? You find a well-balanced casting looks like that. And then the color of everything too. It's look at that spore, bro. It's just
it's so dark. Yeah.
What does that look like? A funnel right there.
Total cycling.
That's like the second one.
We had something similar in that first slide, and it's so weird because it looks like an enormous. It's not, but it looks like an enormous testate with stuff pulled in on one end, because that end is open. That big end is open.
Yeah, that's like storing a bunch of spores in there.
It looks like a smashed trichome or something. Like the color is very trichome-y.
I would agree with that, but remember the trichomes fluoresce like a Christmas tree, dude. I mean,
that's I guess it's kind of hollow, huh?
Yeah,
looks like the buglers I used to eat when I was a little kid.
and it does look like the second one that's had stuff pulled into this end, like it's trying to block something in, and then you see like a nuclei and stuff behind it. The diatom there.
I'm loving the diatom action. And this here, I'm not sure if that's
whatever that is large. That might be an anaerobic dude, like a something in the rotifer range of size. I'm not saying it's a rotifer, but that range of
another piece of fungal hyphae right there.
Predation.
I was thinking
there are some larger anaerobic that.
What's that, Henry?
I was thinking dead stentor. If you look back at the shape of the bugle end of it, it's got that look. Like there's an edoral zone to it.
Yeah.
We should send some Lacromaria inoculation and see those guys go down in here. Maybe.
Could be right with that too.
Yeah.
Size 400. Stentors are pretty big.
So 2 right here is where the bush is.
There you go.
And then 3 will be where that jasmine, that pink jasmine is growing all over the place.
Find him at your sketch. Whoa.
Oh wow, look at him.
Look at that.
He's a big one.
That's a worm right there.
Is that like a full-size worm or a nematode?
No, that's
not a nematode.
That's like a nematode.
That's that. I'm at 100, bro. Here, I'll take you to 40. This I could see him. I could see him on my slide actually.
That's like
okay.
I see a little tiny white dot on my slide.
I was gonna say, damn, that's a huge.
That's him. He's gonna light up.
I didn't see a styler though, so whoa.
No.
They don't like
That's
this
Like,
light,
look at
bro.
that, yeah.
Oh yeah, that
He
looks
does not
dope
like
with
that.
that.
He hates
That's
that.
cool.
Turn that off. Don't be mean. I want to
We're
see
burning them. We're burning them.
He's
Why
like,
are you
ow,
killing the
ow,
guy? Come on, you
ow,
just met him.
ow.
It's like if I met you and I pulled out a katana.
Yeah.
I was just like, hey, I wanted to see what your insides look like.
It's the scene from Blade where he turns on the lights on the fat guy.
Yeah. Yeah.
Here's your carbon, bro. We're gonna light that up.
You've seen light forever.
Look at
Oh, look at that.
You
That's
see in the deep background, you see the biochar, and then everything just stuck to it.
Huh.
Or charcoal, carbon, at least, not charcoal
I
necessarily.
wonder if that's a
biochar,
piece
but
of turtle skin that we're seeing right there.
No, that's crazy.
Yeah, those hexagons, man. That's pretty
Yeah.
cool.
What's that? Cooter or Scooter?
Yeah.
One of them. Look at that piece of carbon, right?
Yeah. Man, look at that. The
beautiful thing.
mineralization.
Definitely.
Yeah, the worms run through that stuff quick. They turn that stuff into black gold pretty quick.
Yeah, what kind of shit
It's
going
really
on?
cool to see that it's actually doing what it's supposed to do.
Whatever that is right there, I can't call it. But
Yeah, I don't know. It's interesting.
we light it up. Looks, you see, it looks like there's like something inside of that.
Mm-hmm.
Yeah.
You know, it's almost like working like an X-ray vision type thing, but I mean, a lot of this stuff. And then look at your saturation.
Yeah.
Under epifluorescent, like everything is deep in the dark ground. It's black, right? Like, which means, and then everything else is kind of absorbed, whatever is glowing.
Mm-hmm. Whoa, look at that.
Oh, dang, what is that? A bug or something?
Carcass. I mean, it's got stippling like it's part of a, yeah, that looks like an insect part. Hey Mo, can you come down? I guess it might be up for you. I'm not sure. The dark brown. There we go. That thing right there.
This big organic piece of matter
up that was coming off of that piece. What looked like an insect part, like it might have been a leg.
Yeah,
or
it looked like a leg or something. Yeah, right there.
That might be an antennae or something right there. Yeah. Might be a soil mite or a springtail.
Definitely got ripped off. You can see how it got.
Yeah.
That's cool because the springtail is so tiny, and to see that is like,
yeah,
like,
that could have been a springtail head that we were looking at just now.
Yeah, that's cool. Shit.
And then a spore.
That looks like
spore
a
next to it.
Antennae. You get
you see the
right
spore
there.
actually sporulating. You see how it's popping?
Yeah,
here?
it's germinating right there.
Which means it's going to start growing.
So that's what we want to see, right? Those rings. Each one of those rings is like growth. So each one, like, is that what I'm seeing there?
Well, you're seeing the spore, and then this little clear area at the tip is an indication that it's actually growing.
Okay.
And usually they're clear until they start absorbing things around, and they pick up that color, from what I understand. Let me jump back to 2. Henry, 200 is where it's at, bro. 200 is where it's at. Look at that. Whatever that is. That's got...
It's full, whatever it is.
Yeah.
It's chilling because it's fat and happy.
Is that like a test date or something?
I was just thinking that, Mo. Like, is that a test? Is that a test date?
You see the red. Whenever you see red, it's algae.
Yep.
Red is algae, you said?
Yeah, because the chlorophyll under this lamp, the chlorophyll shows red.
Okay.
Is that dark field?
Under epifluorescent lighting.
Epifluorescent. Sorry. Okay. Cool.
That man, I would. Maybe
I mean, it looks like a test.
It does. That looks like a big one too. That's a 200.
Yeah.
I think it's huge.
It's huge. Yeah.
Yeah.
So.
Wow.
That's dope. What is that?
It's another one of those guys we were looking at earlier, trying to figure out what it was.
Is that Arcella of some sort? That's a big mouth, though.
No, it's not an Arcella, but it's a test date.
That is nice.
Wow.
Yeah, it takes a little determination and a little craziness to get here. I'll tell you that. You gotta be a little nuts.
Well, crazy is where this group operates. So.
I wasn't willing to quit. I'll tell you that. I was like, God damn, I'm gonna get this. I'm gonna figure this shit out. So one way or the other, I'm gonna figure out how to water my damn plants without having to water.
Good for you, man. Yeah, that's a big deal. Wow. All right, you want to jump to 3? Look at this guy. Yeah, this sample's nuts, bro. I could be here, and I actually
We'd be here all night.
I know, dude. It's crazy. Like I could set up a damn camera and just fall asleep here, wake up, and keep staring at this.
What are your thoughts on this? This is like,
yeah, what is
second,
that?
This is the second or third one I see. It's aquatic for sure, obviously, but I mean, I find this in ponds and stuff.
What did you say it was?
Aquatic for sure, meaning that
aquatic.
This you would find in a pond versus
right.
a compost.
You've got that opening
rectangular,
like a centaur, but it's rectangular.
Yeah,
and that looks. I mean, it looks like a bunch of bacteria on the inside.
Yep.
Sure. And then
does it have a purple tint to it?
Wall though.
The purple tint has to do with the lights and the way they're bouncing off the objective, but this is the reality of it every time. It glows like that. So whatever that's made of is somehow there's a compound in there that's excited by this lamp.
Mm-hmm.
Do you think it could be a fractal piece, like a very small fractal piece of a plant's root, like it maybe like one of like the hairs in like the rhizosphere or something like that, that like the bacteria is traveling in?
Those would be bigger usually, but
typically when you see a root, you can identify the platelet pattern on the root.
Even if it's like microfractaled, like just like a little.
It would have to be smaller than a couple of the scales. They look like kind of like scales patched together.
Gotcha.
You know, I know you've seen some of those AMF photos.
Shit glowing that fungi right there, yeah.
It kind of looks like watermelon tourmaline, but it has a very distinct platelet pattern.
Okay.
Spore right there.
Even it would have to be smaller than the plate.
That spore looks like it's also germinating, Mo.
Gotcha. Oh yeah, here we go.
Yeah, there's the nematode.
There's a nematode.
We haven't had so far as a nematode.
The bacteria eating. So it's full.
It looks like.
Yeah, he's got lips.
Yeah, basal bulb right there.
You can see you can see that. He calls it the basal bulb. I call it the palm tree. It looks like a palm tree when it's pumping. But yeah, the basal bulb there. They've got all the bacteria. You can see the bacteria in it.
See the bacteria in the gut.
Botox lips is bacterial.
Botox lips. There's a dower. There's a
dash. I thought there was a dower up there. Yeah,
dormant one. So look at the size difference, right? I mean, that's massive.
Holy shit!
Size.
Yeah,
and he doesn't like the light. We can
nice tell on that guy.
Piss the nematode off. Just do that.
Oh yeah, now he's moving. Look at him.
Yeah,
what the fuck?
Is the nematode? Is that that's in the successional, like the stage part? That's like the.
They're the whales of the soil, if you will.
Yeah, that's what I was asking. That's like they're not the minnow, they're like the whale.
They're the whales of the soil. Yeah.
Okay.
Those guys in worms.
Okay.
Yeah. And if you don't have them, your soil does not respond the same. And the same with the testates. This is all necessary. You got a testate there. You've got another testate. I can't see
predatory trophic levels there.
You've got a tiny testate there, and then take note of the diversity in the testates that you have. Dude. I've seen so many different types.
At least 6 or 8 different types of testates.
There. So here, although the star of the show is the nematode, we've got a dower here, testate, fungal strand.
Yeah.
I thought I saw fungal strand, but and then whatever maybe, this,
up higher, it might be.
Could be.
Yeah, like that up there at the top could be a fungal strand there along that aggregate. It's hard to.
So you could do an extract from just take a handful of this, put it in water, 2 gallons, 3 gallons, and then provide another side of your bed with this level of biology.
I buy an extract of this stuff.
Fire. There's another testate here. This could have been a testate, might have been, but probably was a testate. Here's fungal hyphae up here, fungal here. I mean, my eyes are just jumping everywhere. I mean, this is just going. This is hitting it quick at 200, right?
Yeah.
So I will definitely go deeper in this, and then I'll start doing different things to it. Start feeding it. Let's wake it up. Let me wake things up in here with.
That's another testate there. That pink.
That right there. Yep.
Yeah. Okay. Yeah. Okay.
Yeah, for sure.
Trying to identify. I'm trying to.
Yeah. They have that. They have that shape to them.
Yeah, they'll look good in most houses.
Look like a pumpkin seed.
Yeah.
I'd like to see. I wonder if I
miss Virginia and I miss the water.
There's plenty of water. You guys could get in it.
Yeah, well, tell me about it, man. I'm out here in the desert, so we don't got water unless you pump it out the well yourself.
Yeah, Adam, you get your hands full up there. Look at that thing, dude.
Look at that guy.
That is like a beautiful picture of some type of encysted microbe.
Is that
When
so
you
you
get
talk
out
about
there on
in the
East
middle there?
Coast,
Yeah,
Yeah.
sorry, the cyst there. It's like a double cell wall.
Yeah.
It's just kind of like, I'm good. I'll wake up when
Okay, that's a double cell wall. Okay, I
you see
what
that.
you're talking- Yeah,
What's-
yeah, yeah.
What's
It's like it's got a baby inside of it.
Yeah, it's
Yeah, I have to commend you, Josh, because
Yeah.
you've
testate.
you've made something
Testate.
truly exceptional in the
Thank you, man.
microbial context.
Yeah.
Aggregation,
Use it to like
aggregation.
guide that
Those
process.
little
You just
aggregate
like
pieces.
Yeah, hopefully it
you just
stays like
nailed the
this.
environment so well from an intuitive macro level perspective, which is like no small task.
4 testates per field of view.
There's 2 testates right there.
There's a broken testate at the top.
There's diatom. So you've got here.
Yeah, that was a testate. Up
2
if you go
testates.
up, I'm
third
sure that
testate,
was
4th
the
testate.
like half digested.
Probably
to
a
the left.
testate. I mean, it's
Holy
like
hell.
probably a testate. We're just like, gung, 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 gung.
Does a
is
testate
really
have
raw.
to have something inside of it to be it, or does it just have the shape?
So the test is the shell on the outside, and there's an
Okay.
amoeba that lives in it. That's why it's called a testate amoeba. So the
Gotcha.
test is the outside.
It's like a hermit crab
And then-
almost.
Yeah, like a hermit crab. That's what I was
They
going to
don't
say.
find a new
Plan.
shell. What they
Look at
do is
that.
they actually grow a new shell once they're full, and then they populate it with some of their nutrients and their genetics. So they'll build out another shell in order to split. But in a way, it's like they build out the empty shell and then they populate it right away. So it's a little bit hermit crab-like.
I'm going to jump to number 3 because I could be here easily.
No, we'd be here till 2 o'clock in the morning.
Sometimes we're here at 2 o'clock in the morning bullshitting, bro. You know what I mean? And
Don't tempt anyone, Richard. We can go right into the Australia.
Hey,
Is the room climate controlled? Or I guess it has to be, but to what degree is your climate controls? Like you got controllers for humidity and temperature or?
Correct. Yep. Everything is dialed in. It's it stays like today. It could be like right now. It's 12 degrees outside, and it's 75 degrees in there. It's 59.3% humidity. My VPD is 1.23.
He said VPD.
He did say VPD.
Damn. Damn.
CO2
Next we'll hit him up
CO2 is
for
350.
His PPFD.
PPFD is 0 right now.
Yeah, I would say that's actinomo.
Actino, yeah, agreed.
Yeah.
There's the Actino.
This is number 3
Yeah,
now.
that's
Yeah,
sounds like
3.
AC Infinity to me, Steven.
That's a pulse meter.
This is the pulse meter, okay.
Yeah, the Pulse Pro. They sent it to me for free.
Yeah.
That guy.
So all I gotta do is mention them every once in a while. So there you go.
Texture on the outside of the thing.
So
Save
you did your part
10%.
for the week. Yeah. Yellow Skunk Farms.
That's right. That's right.
Yeah.
Save 10%.
That's like pollen or something.
Great seeing you, Derek, man.
Hey, Derek.
Good night.
Derek.
Good seeing you,
That
Derek.
could be pollen, Mo.
Take
Yeah,
care,
I
man. Take
agree
care.
with that.
What do you suppose Derek's
Because
temperature
Josh, you've got
was?
both male and female plants in your setup, right?
Yeah, we did. Yeah, I had one male. We're doing this run to do a seed run.
Oh,
We're
you're
doing the
seeding,
seed
Wondering
right?
if that might be pollen right there.
Could be.
That could be pollen. That's very possible.
Kill it.
Very possible.
Lit up. You got good. This is actinobacteria that see. So a lot often mistaken for fungal because it's a hyphae.
So you say that's not fungal strand there?
No,
that's
actually bacteria.
That's
and it's funny 'cause it used to be called actinomycete, so it used to be like a fungal-like, but you can actually see the little bacteria lined up inside that filament.
Okay. Yep.
And when you
Yeah,
walk
I
into
do see
your
that.
when you walk into your grow and it smells just like soil.
Yeah,
That's what it smells.
We still love them though.
This is
Oh, yeah,
what
they're so
does
good,
that.
yeah.
This releases those hormones that make everything smell like earth.
They're great.
Okay.
And
They're
it does that to call in all your anthropods and all your bugs so that your bugs can walk past it and take away spores and take it to the other side of your system and start growing over there. It's beneficial.
Oh, that's cool shit. Okay.
Yeah, but that smell you get, that's design, bro. Like I heard the camels can smell that from miles away. The
Oh, I'm at it smells fresh, dude. Like when you walk in the room, it's
bet,
like
dude.
totally different smell than outside. Totally different.
Oh yeah, I bet.
I mean,
All this
look at it.
biology going on in there.
But again, here everything's settled to the bottom. And what I'm gonna do is rehydrate it. It was a little dry, but there's fungal there. There's a hyphae there.
Okay.
There's a
That's what
testate.
fungal looks like there. Okay.
There's a testate right there.
That's fungal.
Yeah, that's
yeah,
fungal.
That's a piece of hyphae right there.
Yeah.
And you're looking at it you're looking at the septate.
Septation, clean broken
these
in.
Little these little lines here.
Yep.
You're also looking for. It's not consistent here because I might have crushed it.
That's a crush.
Otherwise, they're very even and uniform. As far as thickness, they don't get too wonky or crazy. They don't bow out or anything like that. And then also the color. You see that color? That's money, dude. That's spot on. Those are the colors you want. It's a nice
fulvic humic color.
It's probably four nanometers easily, right?
Yeah.
Which means it's thick, beneficial. So that is, other than that crush right there, that's a nice example.
So will that grow?
There's also the texture. It always looks like glass, like a crystalline tube. There's some kind of a texture to it that you're like, that's fungal. Yeah. Come on, babies.
Say it to you, Annie. Yeah, so just know that's when you're under scope, bro. When you get your scope, Adam, hit me up, man. We'll run a fucking session.
Oh hell yeah, dude! I'm excited, man. I'd love to.
Well, Monday night, bro. This
Yeah.
link is always available, bro.
Yeah, we're here every Monday for like 5.5 hours.
Nice. All right, cool.
Bong rips allowed. Look at this thing.
Yeah, no shit, right?
Oh, he did. That's all you had to say for me,
man. Bro, man, don't hold back, bro. This is what you waiting for, man. That's it. Yeah. So number 3. So I'm gonna rehydrate this again. I learned that just in testing things. Certain castings that I've tested that were just ass, and then
more turtle skin.
Yeah,
it could be shrimp shells.
Shrimp. He's got shrimp in his shit.
Molting.
Too.
Okay.
Molting
you
shrimp.
have a mono shrimp or ghost shrimp?
Cherry.
Okay. Okay, cool. So you got the Neocardinia.
Even in this.
I don't even. What's that?
The little small red.
Big old spores in there.
Yeah.
That's not a spore, bro. That's Arcella.
Yeah.
Scientific terms.
That's a testate.
No, I said it had 2 spores in its belly.
Oh, yeah, it's spores.
And it does look like that.
Yeah, it does kind of.
Arcella. Oh, it's
look at that.
Wow.
Yeah,
it's like a donut. This is like a donut, but I don't know. They have a hole going all the way through them. No, this is like a door. This thing's packed. Let's do this real quick.
Arcella are crazy.
Arcella are cool, dude. They're like, what is that? A quality thing you find on rocks, bro? They're squishy. Trip.
Or have your brother help you find it. Okay.
Yeah. So number 2, hands down, right? Is your extract for sure.
It does kind of look like a closed anemone.
That's what exactly. Thank you, bro.
Yeah. Like when the tide goes out, they don't have their tentacles out.
Yeah, out there, the West Coast, bro. California, Mexico. You see them on the rocks.
Yep. Or you step on them and just whoops.
Yeah.
I have a question on that actually, because we saw those 2 spears inside there. Does like the testates, do they go through a state like the way like prophase, anaphase, and metaphase, like cell structures go through? Do they go through that as well? On when they produce to get out of their shell, when they separate?
These things build their shells, bro.
There's a few different ways.
Full, and then they'll build a shell mouth to mouth, and then they'll populate that shell, and then that's the split.
That's the split. Okay, so that's how they produce.
Yeah, that's how they reproduce. Yeah.
There's a few different ways how they actually manufacture the shell.
Yeah.
What is that?
Some of them acquire shit from the external environment and assemble elements together.
Clear on that one end too, Mo. It looks like that might be germinating.
I've seen that particular spore on every sample. I have to go to my key for my spores, bro, because
they all got that little tail coming out.
Others create their
I'm
own.
waking up.
That clear, that's the germination right there. You see that?
Yeah.
That is it growing now.
Okay, so that's actually the rings that you see. Those are growth rings.
No, you see
no, this is a spore.
You see this kind of clear, transparent here?
Yeah.
That's it growing.
Okay.
So before that, it could be
a hyphal extension.
Right, exactly.
Yeah. Okay, so this is a spore that we're looking at, and it's growing right now.
Yeah.
Right.
Germinating. Okay, cool.
Yep.
I gotta know that because I have a lot of different types of inoculants that I use that are supposed to have a certain amount of like spores inside of it. And I want to test the stuff. So when I get my microscope, I want to be able to tell to see how many spores it actually has.
Yeah, because
yeah, you want to know that.
Yeah, the label's gonna be like 5 billion spores per.
Oh, look at that! Look at there.
What is that?
Yeah, all that shit they keep advertising, billion spores per all that stuff. It's like, really though?
That thing's got a telephone line running too.
Or when they say it's 1-to-1 fungal to bacterial, and then you're like, yeah, there's one spore and 5 bacteria.
Yeah, exactly. So what's that between these two things here?
So that's a piece of cellulose right there.
That's cellulose,
yeah. And then there's a piece of actino that's kicked off over here on the other side.
There's another piece there. So cellulose is being broken down. It's being decomposed.
We know that, Jeremy, but it sounds good to the ignorant. So,
so and then Mo, when you flick off epi, there's a piece of actino over to the left of this, right there in the background.
I see it. Yeah.
Oh, yeah.
Stretching.
Yeah, those long spirals you find, those are pretty much always going to be cellulose.
You see what he's talking about, Josh, right? This
right here.
That's actino.
Yeah, have you ever looked at the comment section on the product reviews for predatory nematodes?
Huh?
No.
Like if you look in Amazon at predatory nematodes and look at the comments, you'll see people are like, 1 star. They were fucking dead.
Yeah, they're fucking dead. Yeah, that's why I always see it too. Yeah.
Then you'll see one person that comments like, I actually have a microscope and yes, they were dead.
Man.
All right.
I called the company and told them and showed them the picture and then they replaced them.
He's right on that.
But it's the problem of trying to get the shit to you that usually kills it.
Potential.
Well, with Amazon,
is that an end of a nematode? Is that like the half back part of it?
No, those are the buglers.
I don't think
I
so.
think that might be a stentor. That's what Henry was talking about earlier.
Yeah, Henry thought that potentially a dead stentor.
Yeah, the way it widens out makes me think it would be too wide to be the tail of a nematode. But.
That's that. Yeah, I wouldn't call that a
nematode.
No. Well, they've all been symmetrical like that too. Like whatever it looks like a thing. Like you're not going to break your nematode exactly the same.
No.
Yeah, it looks like a piece of a nematode for sure to me.
What's your water source, Josh?
Well, it recirculates.
Yeah.
Think, right?
Yeah, but he gets it from somewhere to replenish.
He's not getting it from his river or anything, right?
I thought in the YouTube it said like a creek on his property or something.
Oh, the replenished water is coming from the well. We have a deep well.
Okay.
We have a deep well.
So you can get aquatic shit from a well for sure.
I drink the water that I've
That's okay. You want to get them out?
There's a spore right there.
Look at that spore.
How deep is the well?
It's 126 feet.
There's likely not very much aquatic life in the well.
Have you tested it for biology?
But it's basically fodder for aquatic life in an environment that's already aquatic.
Yeah, I'm basically just topping it
there,
off.
Mo. It's winding its way through the back right there, holding that piece together.
Well, let's light it up.
Oh yeah, look at that.
Can you see the spores in there?
It looked like there might be.
Yeah, I saw one in the corner.
Oh yeah, right there.
No, let me light it back up. It's right here.
Right here.
Right there. There's a cluster of 3 spores right there.
Okay.
Oh, okay.
How do you see that? Like, how do you?
Yeah,
there's actually more. There's 5 spores there.
It just looks like humic stuff to me.
Mo lives on this thing. That's how
right there.
Yeah, that's awesome, Mo. Wow.
I can't see them, but.
And then so like on the I guess my left, that round spot, that would be a testate.
That could be a cyst. That could be, you're talking about here?
Yeah.
It just looks like a brown splotch to me, Richard.
See, like there's a darker
brown splotch. There you go.
Like I see him now.
Maybe that's because you're on your phone, dude.
No, I'm not.
with my
This thing says buffering. He's like, "What's that buffering thing?"
Why is everything a rainbow wheel across the entire thing?
That's like a cyst of some sort. If I had to guess, but and I make that distinction by the way it lights up. Just my work with epi, and that's just my observation that when I hit things with epi like that, and I see them light up, they tend to have been alive and now or an egg. It could have been an egg. You see, it looks like there's something that ruptured maybe, or there's something happening in the middle of that, right? That doesn't look organic to me or live, because usually if So if it's an amoeba, it would be all clumped up in a corner or against the wall usually. Here if I was able to get into that, something's
and Mo, it
obviously
almost looks like
happening
that
there.
spore that you showed that spore may have germinated and might be this piece of hyphae we see over here holding this entire aggregate together.
It does look like it's swimming through there.
It does. Like it goes up and comes down, and
yeah, it's up on that left
Yeah,
shoulder
that hyphal structure is wound all through this.
Oh yeah, there's hyphae all in there.
It's going all the way through it.
You see what he's talking about, right?
Just
It's going
take it over.
right here.
Yeah.
Looping,
Okay.
coming over here, whipping out. It's like a rope.
Whoa.
So it's decomposing this, bro, right? It's just tearing it down.
Whoa.
Taking nutrients
You broke
from
it.
it. Look how lit up he is, right? It's got all this to feed off of. Look at he goes down here too, right? You see it at the bottom. Why is he fluorescing here? Why is he's probably a little higher up on this piece of organic matter where my epi, and that's at 400. If I take it to 2, I'm gonna get a better view. Because 400 things get a lot wonkier with epi, but you can really see this thing as I do that, and the cluster of spores looks black up there.
And like you can see that black winding right through there through the middle too, and coming up towards that spore, and that's where it comes out on the side over there as well.
And he's about to he's bouncing out down here.
Yeah.
Looks like it was already here. I might have done this. I might have caused this by mixing it, right?
Wow. Okay.
Yeah, that's cool.
So it's just traveling, it's moving.
So do you think it's trying to like pull those two pieces together eventually?
It's decomposing them. And
Mo may have severed this piece down here from this main clump, but that fungal hyphae right there is definitely that spore germinated. It's pulling this macro aggregate together. So like there's some micro aggregates around here. You see a piece of cellulose between the two right there.
Yeah.
So the bacteria will make the micro aggregates, then fungi will pull those together in bigger macro aggregates.
This is a this is a microscopic look at decomposing organic matter, bro.
Yeah.
So this might be a pretty ignorant question here,
Nothing's
but like,
ignorant here, bro.
Yeah, man.
So,
well, some of us can be, but
cellulose between those two things that are connecting it, like those aggregates together, like that you're circling right now.
Yeah.
What's causing those?
So that is cellulose. That's formerly plant material that's been broken down. That tight spiral like that is characteristic of cellulose.
And then look at the bacteria dancing around it. If you're asking what's causing
all that right there, yeah.
Did you get my explanation on how?
Dude, there's the other end of that fungal hyphae right
there. Is always dead plant
the switchblade ninja enzyme. Did you get that white paper from me?
No, I didn't.
Bacteria goes up to organic matter, releases enzyme, which are like knives, bro. And these knives just start chopping shit up. Or you know what? Josh had a better analogy. When he ate a steak, his fork and his knife are his enzymes.
Yep.
And as he's tearing that steak down, he's absorbing it, and that becomes But that's how things get broken down, even at a bacterial level. Because if I saw bacteria around it, I can see bacteria there. You probably can't see it, but that little dot right there where my hand is, my thumb is pointing. I mean, I'm gonna point at it with a little finger right there.
Yeah.
Bacteria there, bacteria here, bacteria here. They're
This-
all working this shit, right? They're tearing it all up.
All these little spots on this aggregate right here are bacteria.
Like,
cellulose is like the fibrous material of the plant, so it's going to be the slowest to break down in general.
You're right.
It's not like the sugars
an awesome
that are
macro
inside
aggregate to look at though. With this,
That is an amazing
Yeah, it is.
Man, dude.
Looks like a continent, like Africa or something.
It does.
Greenland.
Yeah, it's Greenland.
And then don't sleep on the test state, just
conquer
chilling
this
over
shit.
here.
They're mining the rare earth
chilling over
minerals
here on
in
the side,
Greenland.
right?
That's
Yeah.
it. Like, I got this. Go get some nutrients.
Fungal.
How you doing, Rajani?
That's cool as shit.
You want to hear something cool shit, dude? I think it's mind blowing to me. So this test state right here operates, and I'm giving you rough numbers off of a 15-to-1, like a nematode, carbon to nitrogen. These bacteria are a 5-to-1 carbon to nitrogen. So these predators will eat these bacteria, and they need to get to a 15-to-1 nitrogen to balance themselves out. So in order to get to 15, they need to eat 3 bacteria.
3 of them, yep.
Right, but if you do the math, how much nitrogen is that? That's 3 nitrogen. So what they poop out is the excess. It becomes the available nitrogen to the plant.
Yes, yeah, and it's available. Yes,
right. And so they're constantly feeding to keep this balance within their bodies, Go find out what our carbon to nitrogen ratio is. It all is relative somehow, bro. It's a trip, man.
Yeah, hell yeah.
Ours that
Yeah.
Or that poop is
I
food
eat
for the
plenty of
bacteria
carbs.
again, and the plant just
Oh,
eats
this thing
the
has been
bacteria.
slowly shifting this way that I circled here.
Yeah.
Slowly shifting this direction.
Well, you gotta keep in mind that this slide at some point is gonna start drawing.
But yeah, but
out.
nothing else is shifting, but that
So you're
was.
saying so you want to go in deeper?
Yeah. Oh,
200 and 400.
We have the technology.
Yeah, that guy right there, that little group right there was moving around.
It's Iceland, bro. Come on.
Everything's just jittery, bro. Everything's just.
I know
buzzing.
everything's fucking moving.
You've got yeast here. You've got yeast here. So, I mean,
Lots of bacteria.
Lots of bacteria, right? Yeah.
Bacteria. Doing their little jiggles. And you can tell they're happy. Look, those ones are horizontal gene transferring.
Yeah, there they go.
Oh, cool.
Where do you see that at?
There's a couple.
See those three?
Together.
Let me draw.
I want to see some microbe sex.
You see this right here? Micro porn.
The way they orbit each other.
There's a microbial orgy going on there.
And make contact.
We should make a podcast, microbe orgy.
Yeah.
That'll get some views.
That'll get them.
Yeah, that'll get the views right there. It might not be what they're looking for, but it's gonna get views.
Yeah.
You see that little rod?
Was it
there?
Arcella that you were showing that one time, Maurice, where they were like super mouth to mouth?
I don't know what those were, but they were at it, bro. That was cool.
Yeah, the bigger things, it's crazy to see that stuff happening.
I got some pretty good soil porn. Yeah, that's fucking for sure. Look at this here. What's happening?
Yeah,
here?
whoa.
What's that? Why are we?
But you want that because that's the
that
transferring of information.
Of course, yeah. And you're creating a Frankenstein, right? They're swapping and.
Kind of like what we're doing here.
Swapped around and.
Look at that. Bro.
It's so much easier to relate it in your mind when you relate it to porn. You know, like I just get it a lot more.
It makes sense. Makes sense. Look at this.
Like a testicle. Got it. All right. Let's move on. Let's go.
Naked amoeba. Got it.
Yeah, naked amoeba, right. Yeah.
You gotta make it relate somehow.
That thing's really cool looking, Mo.
Yeah, whatever that is, it's not. It's closed. Shop is closed. But then these guys here, look, there it is.
Could be a naked.
And this is you want to see this, right? You want to see this in your sample, bro. Right? This is again what we learned from Matt. This is the stuff you want to see, bro. You want to see them. This is a party, bro. They're mingling. They're swapping. They're happy. They're buzzing. They're cool. If I were to change the pH right now,
roofies.
What you're saying is that they're all getting along, basically.
They're all getting along.
Right.
Again, like we talked about earlier, all these samples are very, very calm and just they have like a zen to them. Nothing's really frantically moving around, trying to go, holy fuck. I gotta survive, or I gotta reproduce right the fuck now.
It's kind of like they're all happy, they're content.
They're in balance.
They're
and
satiated. They're also satiated. There's no one starving, and there's no one. You can have starvation, but you can also have an excess of an input that then causes them to be like, we have to process this like it's do or die.
Right.
Gold rush.
And too much would bring out predators as well, which I'm not seeing a lot of predators either. Like a whole bunch of other things, and they're trying to eat up all the bacteria or anything else really quickly. So, like, it seems like it's
No, and that shows that the predators have found their equilibrium where they're constantly having the food available, but they're not having to outcompete each other. They have their territory, as it were.
Is a nematode a predator?
There can be predators. Some of them are.
So a predator nematode eats other nematodes.
Some eat bacteria, some eat fungi, some eat roots.
But it is a predator because it eats bacteria.
All of them are, in a way, a predator in that they're eating lower trophic layers. So yes, like a wolf is always a predator to rabbits, but when we say predator nematodes, we mean the ones that eat other nematodes.
Oh, I mean, that was a bad predator with carrot though too. So I mean, they aren't innocent here. All those carrots are out there.
Yeah, it's true.
Care for their lives.
It's a rabbit eat carrot world out there. So.
Stuff, yeah.
So what's that in the center there?
Is that a testate?
That is some type of, and it's not.
Is that a shape like a testate?
It's got a mouth.
Oh,
Might
I
be
see.
naked though. Maybe it's just it's kind of malformed. So
I would agree. I would agree that that's a
That
naked,
seems big.
insisted naked because
Could be.
that would be too. I mean that I don't know. That doesn't look like a testate to me.
because it's got a mouth on it.
That's why I like where I circled right there. It looks
But
like
it could
there's
just be an opening. It could just be the opening for the cyst.
Yeah, it could be.
It doesn't have that defined flat like.
And it also it doesn't have that thick double wall that
it's not stout enough.
does either. Yeah,
it's kind of and it's very large. It's exceptionally large.
Yeah.
You know what's really cool, Josh, is that you're gonna go to your beds now and you're gonna have an idea of what they look like on the inside.
That's gonna be stupid.
You're gonna break out a dictionary and go, "What the fuck were all these words they were saying last night?"
Corner there.
I don't sleep much anyway, so
there's that spore we were seeing earlier.
Yeah, all I got was testate.
Spore. That's a very iconic spore.
If I get a fucking
You've seen it in every sample too.
It's gonna be a bad thing, probably.
You bet you gotta have your bong next to it, bro, and you'd be 4 hours later, like, what time is it?
No.
Hypey right there.
I can see myself missing my morning, my morning video, like I sleep in or some shit.
Hey, you're extremely consistent with that, man. That's dope, dude. It's every morning you fucking.
Once you start throwing those microscope slides on those videos, man, dude. Yeah, that's the future right there. People want to see that stuff.
Yeah, I like that. You know, I'm. God, it's come so far and so fast. It's just,
yeah, the tech
it's going to go to like
or maybe Actino right down there.
The next stage of this is growing food. I want to grow food, and if you guys, like I said, can teach me how to see this stuff and then really dial it in to where like you can really grow nutrient dense anything, not just medicine but food.
You're doing it, man.
Hey, say
bro. You're locked in. You should put some rice in this thing.
I'm not sure what
that looks like an encysted amoeba right there, though. That
could be. It could be.
You could crush it with some rice.
Not that that's double
like
wall.
the most productive thing to do, but
where it looks like a fried egg
on a large scale, though. In a greenhouse, on a larger scale, to where you can actually build a pond like an aquaponics. I mean, they build huge aquaponics systems, like. Why not build a huge? You only have to build it once, and watch it get better and better and better. And there's no maintenance. The only maintenance is going in there and making sure everything's flowing right, cutting down what's growing, keeping it in check.
What would be cool, Josh, is when you were talking about it.
That'd be awesome.
When you do this again,
a test,
you would do it completely different, right?
Yeah.
And God knows what you'll put out that way, but look at that action right there.
That's crazy looking shit. That's some alien looking stuff right there.
It is.
Testate, maybe a naked.
Yeah, it could be naked. This would be fun to see after about 3 or 4 days. See what
I can do that.
Yeah, it's like you see stuff moving around inside this.
Oh, this shit moving in there, bro.
But it is like Mo was saying earlier, like it's amazing how balanced humans are to this whole environment. Like healthy food, healthy humans, it's all the same ratios and elements, microbes. That's why I'm here.
Mo, can you go back to that big
when you say we're all connected, that's what we're talking about. We're all connected.
Yeah, absolutely. We're all the same. I never felt closer to a plant in my life.
Yeah, that's a trip that plants are even further away from us than fungal are, because fungals breathe at least like the same air that we do. And extract like CO2.
Jeff, thought it was
way more closer to fungi than we are plants.
Yeah, exactly. That's what I'm trying to say. Yep.
Plants are crazy off the deep end.
Thought it was down. Yeah, I don't know, but
we've
seen like 5 or 6 of those tonight.
Oh yeah, we've seen one on every sample so far.
That spore, that specific.
What that little spore? Yeah,
the little pine cone spore we've been seeing.
Yeah.
In every sample, yeah.
And Rich has a poster on his wall, so I was just, if we could identify it, that'd be kind of cool.
That'd be awesome too. Yeah.
Probably easily identifiable.
Just point his camera at the poster.
Look at this organic matter here, but then look inside of it, right?
I can, but it's kind of far away.
Oh, that's cool.
Like what? There are things.
Cell walls in that, yeah.
It looks like it's skin.
Magnetism are you at?
I'm at 200.
200, okay.
And as you get more time, just seeing
200 is cool. Look at this cluster of spores right there. You see it?
That's so many.
Right there.
You see that?
Yeah. That's awesome.
You really start to see how the microscopic world looks a lot like the environment that it comes from. Like it's a direct reflection.
Absolutely.
Yeah.
Like
Micro and macro.
you see like a forest when you look at the deeply fungal, forest soils, you're like, oh, that density matches the density of the plants in the big
There you
world.
go.
Yeah, it's not unfair to be like, that is the whale of this ocean.
Yeah.
And I'll point this out, bro. Most slides that look like this do not look like this.
Right.
I mean, all of your shit settled at the bottom, organic matter at the top. Most of the time, when I look at a tube that does that, I'm like, this is gonna be quick. Right? Look at we've been picking apart this and look at it. It's
So you're not seeing a lot of humic substances.
I'm not seeing a
is
lot
it
of
pretty
humic
clear?
substances. It's
Is
very
the water
clear.
They're very clear
It's
in the middle.
absolutely
Yeah,
clear.
you got organic matter at the
clear.
top. Sand
Yeah,
at the bottom,
so that
clear.
re-emphasizes
see
that
that?
this is less of a traditional what we would call a soil system and more of a semi-aquatic system that you would find along like creek banks.
That test.
So this,
his humic fulvics are probably leaching off in the water that's constantly being circulated around.
And it's going somewhere in your system,
well, that's
right?
a good shot right there.
That test date.
It looked
Test
like
date.
great aggregate there when Mo was showing it. Crystal clear water.
Crystal clear water.
So what do we look at? Test date?
But in the sample, what you were
right there. This might
just showing in your camera?
Yeah, that's his sample.
Crystal clear with some aggregate.
Yeah.
What does that mean?
It means it's not just exploding in the water.
It doesn't have some of those mature, longer chain humic and fulvic acids that will make that a tannin color and increase the.
So is that you wouldn't expect to see what you're seeing? In other words, like you would
like the amount of biology.
Yeah, a lot of times when things are clear,
let me clarify.
Very empty biology.
When I look at this before I even put it under the microscope, I'm already thinking this is done.
There's nothing.
This is going to be trash.
This is going to be a trashy sample, right? But look how much time we already have invested in this.
Bacterial feeder, Mo.
Different level of balance. Yeah, he's just gonna food coma, bro.
Is that a naked on the top right there? Is that just a little piece of sand or something?
Top right, top
corner there. Yeah, kind of now centered.
No, that's a piece of something. Definitely not naked.
Particle.
Though. Yeah.
I will check that out here in a few minutes, Steven. I'll jump on my phone and check you out. Whoa, was that a cluster of yeast back there, Mo, that we saw?
Yeah.
That right there? Well, a cluster of yeast just hanging out, doing their thing?
No, I wouldn't call it yeast, bro. I've seen that before plenty of times, and I can't call it.
Okay.
Oh, I'm not a cannabis guy, but I had a question, Josh.
Yeah.
Do you have I guess a couple of you guys on here have said weed plants are very finicky about their water. Do you not have any mold problems with this much water?
No. Well, they're talking about bud rot and
root rot,
bud rot. A lot of cannabis growers, they once the flowers get into week 8, week 9, almost ready to harvest, you can get moisture in the room, humidity can get high.
No,
that's what a lot of people are concerned with this system. Is overwater and too much water being evaporated out into the room. And I deal with that with just an oversized dehumidifier. So I just have a dehumidifier that's rated for 1,500 square feet.
What
are we looking at?
You got a spore.
500 square.
You got it. Looks like a spore here that's sporulated. It built up another spore here.
Yeah.
Built up another spore here.
So that's mycelium then, because it's catching things. So no, it's no longer a hyphae. It's mycelium.
That's mycelium.
Yeah, all right.
Bro, you're kidding me, dog. Damn.
You're killing me, Smalls.
Did you just grow a penis on my mycelium? Here you disrespect my mycelium. I grew that myself.
Well, then also, Jeff, if you were growing in container, you'd be worried about fungus gnats. Once your soil got too saturated, stayed saturated for too long, you'd be crawling in fungus gnats.
Yeah. And that's a good thing too, because I was doing a morning mist every morning. So I was morning misting, just trying to replicate the dew, like a morning dew in the morning. And I noticed real quick that was the wrong thing to do, because it was just holding too much moisture in that top organic layer, and then fungus gnats would just boom. I get an explosion of fungus gnats.
Yeah, they blow up.
Because you don't have cyanobacteria in your dew. Come on, man. Like, be like nature more.
Yeah, no kidding.
But
I
you
stopped
never
doing that, and everything went back into
you
balance.
do
It's
have a
amazing how
natural.
elegant nature is.
Yeah, you have a natural mist just coming off of your ecosystem there.
Yeah.
From having the waterway and everything, the natural evaporation that's occurring.
Yeah. Cyano would really punch this system up even further, though, as far as the amount of energy and nutrition in it.
Wow. Bro, look at this shit moving inside of it, bro. That's so
that's
like, what the hell?
Get this with the Epimo, if you don't mind.
Dope.
That's a good find there.
That's insane. Look at that.
You said this is 3? Is this soy bed 3?
This is soy bed 3,
this
man.
Is 3.
Okay.
Yeah.
This is what I told you. I told you that I thought that this one would be better, because 3 is where I've seen the most diverse plant growth. Like, it didn't matter like what I've planted in it. It's just been everything that's come out of it has been really good. And
that's
crazy.
You can see the things
better.
Wow. You can see the things here. Let me see if I can pump this up a little bit. And then move it over. You can see things moving in there.
There we go.
Oh wow, look at that. It's like a highway. That's a super highway right there.
Cool.
And down.
That's so
exactly
Rich.
what it is.
Rich, you're saying this is hyphae, actual hyphae.
Holy hell. Look at all that stuff
Super
moving
tunnel,
in there, man.
Like a speed train.
Yeah, back and forth too. And it looks like they're going back and forth.
It's like a 2-lane highway, bro.
They're going opposite directions on either side of the
hyphae. My understanding is a hyphae is like the little segments that we see, but when it actually starts, once it's connected to something else, then it becomes a mycelium.
And then look at the spores. Like it's
plopping
out spores, and then fucking spore turns off into another direction. Then there's a chase down at the bottom with another spore over here.
We never did that. Like this is
no. We never see.
We never see.
I had a record in 4K. Yeah, I've never seen this. This is cool. I dig this.
That's dope.
Well, I love you can see the movement inside, man. That's just,
right?
That is like everything that we're told, but we're looking at it now.
Yeah.
Now I believe it. And now I believe it.
It's like seeing
microbes European. It looks like they're on the wrong side of the road.
Yeah, no shit.
I could be Mexico.
Mexico.
Drive. It looks like they're all going.
Lots of places other than the U.S.
Where's the roundabouts, right?
There's a roundabout right there, just below that.
Can I ask a quick question? Like, if you were to look at an old growth forest, this is what you would want to see. That long fungal strand.
Darker coloration and larger diameter.
Darker color and larger, and
So like those darker colored ones we've seen, they're kind of like a 70% dark chocolate in color.
Yeah.
But they would be like 2 and 3 times that size.
Right.
And you'd see a lot of like the early one we saw where it looked like there was like bumps where every septa was. It's called a bracket connection.
Yeah.
The Basidiomycota. A lot of those old growth forest systems, you see a lot more of the Basidiomycota in those clamp connections.
So in basically other words, this is the beginnings of
you're getting this if you look at this fungal bacteria, you're probably going to be looking closer to your 5-to-1 biomass fungal to bacterial, if not a little higher.
Well, wait a minute though, but if we're looking at hyphae's like white. It's hyphae's visual to the naked eye.
Mycelium. So
mycelium, yeah.
That can depend upon the species.
Okay.
Because the species, this might be a species that has a very small, like this might not be a fruiting. You may never see a fruiting body from this ever. Whereas like the big ones that you see, you're more likely to actually see something that has a fruiting body.
You know what, dude? I see it on my slide.
Yeah.
Yeah, I can.
I was like, yeah, never mind. I see it now.
I can see it on my slide. Yeah, that's dope.
Dancing around.
What's that monstrous thing that was down below this, though, Mo? You just went down. Keep going. Yeah, that right there. Because dude, there's all kinds of shit growing off of that.
Whoa, yeah, look at that. Holy hell.
Growing,
Mycelium,
Yeah, he's on my side.
Mycelium often take on a color of their environment as well. It's not just species dependent.
I'm looking at this whole little area.
Okay, that's good to know.
Slide. That is just a wicked aggregate, bro.
And it's just sprouting.
I think that deserves a bong hit.
Yeah, rip it, bro. Hey, good job, man. Good job, man.
One hit per finger.
You get 2 meatballs, right?
2 meatballs tonight, Dave. Thank you.
You get 2 meatballs.
Oh, wow. Look at that real color there. That's dope.
Look at this,
what the
man.
hell?
This is why my students went nuts on it. It's just
Oh, look at that. You can see all the freaking minerals and stuff, too, on that aggregate.
And then look at that. That was that a spore in there?
Spore right there, and then there's something down here that has a circle of mineralization, but it's open in the middle.
Because he could see your finger on the screen.
No, was that really?
Richard's like right here. He's like pointing at it.
That's from my own visual representation of where it is. But I'll draw it like there. It's right in. There you go.
A little circle thing. It's like a little.
Yeah, but there's mineralization around the outside of it, but not inside.
Whatever's glowing there, that's like an orange color. That's kind of rare. I mean, not rare, but. And then what's happening here? You see that ring?
Yeah. That's what I'm talking about.
Oh, is that what you were pointing at?
Yeah, that's what I was pointing to. Yeah.
Yeah. Okay. Yeah, I have my face on the other side of the.
I mean, you're good.
Look at the fungus. You can see the hyphae inside of it when you do this. Look at there's algae
you have more
here
minerals
too.
growing different colors up here.
Algae or mineral? Probably more mineral than anything.
Yeah, same more mineral than algae. Yeah.
And then look at over here. You're just getting.
When you say algae, is that the type of algae you would find in the water?
You know, algae's got chlorophyll. Yeah, well, you know, cyanobacteria, which is spirulina, diatoms, anything that
large class of organisms there.
Hey, there you go. Thank you, sir. Figure that out. That's wild, man. That sample's nuts.
Yeah.
I tell you that most times, if that was my sample, I would probably looked at that and not even put it under the scope, because when I see things like that, I kind of
you didn't expect to see that fungal hyphae with that.
No. That's the first time any of us have ever seen that. I think because I know
you know what that just inspired me to start looking at mycelium, because I normally am I'm inside the compost, here doing the work at the microscopic level, but I don't usually pull back like that. And that was on my slide. That was a small piece. I could see it on my slide. It's a small piece of aggregate there but I'd never seen that, dude. And I don't know.
If there's something
that was a great for me not to see that. Yeah, that's a great find.
Oh,
no, for sure, absolutely, all day. Look at that. So that's sporulating, right? That's creating spores.
Yep. And look, it looks like there's some here behind it too, Mo.
Oh, we're gonna go deeper now. Watch this.
We're on the left. Yeah, look at all that. Look.
This is the under canopy of it all, right? There's like a mushroom right here popping, bro, right? Look at that.
Holy cow.
Dude.
It's like a little trichome head. It's a little trichome.
Yeah.
Wow.
It's the mushrooms part like that. Yep.
That's the cat.
And then you got more in the background further back. It looks like actino maybe in the background here. The really fine threads right here in the very back there. That right there.
Now we're like deep, right? We're like deep 6 here, boy. We're the fucking and at every level you're rocking, dude. Check it.
Yeah.
Every time you focus, you're going to a different level. Is that
it works way down through the sampler, back up through the sample. Yeah.
Let me take you all the way up, right? So right now we're all the way up. We're way too high to see anything.
Right. Then the first layer you come to
is that big. I'm going down, and then boom, we start seeing the bigger chunk that's at the top, right? So you can think of it like an iceberg, maybe.
Yeah.
The tip of the iceberg, right? And then here, if I hit this, now we start looking at what's up here.
Oh wow. Okay.
Right? Yeah. There's levels to this shit. Then we come back here, and then I keep.
Think of it like a CT where you're taking the slices of it. You're going down.
Yeah.
Each different focal layers.
It's interesting. It's interesting how when we notice the focal length when on a microscope, it's the same exact thing when you're using binoculars.
Mm-hmm.
You know, like when you're scanning
Sure,
across the ground, you can use your focus to keep it in focus as you get closer to yourself on the ground. So it's no different. Focus, the focal point is literally just the depth.
Yeah, and which you're focused.
That's right.
Bro, honestly, I've never seen a piece of organic matter pop like this, bro.
No.
And then look at this. Look at all this. This is probably yeast, if I had to guess, but it's not. Look at here.
Yeah.
We've got actinomycetes going on here. We've got fucking.
Oh, whoa, yeah, look at that.
All kinds of stuff happening here, dude.
And this is the second piece of organic matter we found that is the second piece of macro aggregates.
All here in number 3. So you called it, you called it off rip. You called it.
But then still looking at sample 2 was so thick with so many things too, though. Like they're
but the moisture level of sample 2, the moisture level of sample 2 was greater.
Right.
By a lot.
Right, and we saw all the test dates.
And like Jeremy said, bro, that's where they listen, microbes swim, bro. So that's probably where I don't know, man. I don't know.
He's probably got more free nitrogen being made available in sample 2, but in sample 3, he's getting more of those secondary metabolites. He's getting those fungal byproducts. He's getting more complex things being fed to his plants.
Take it to 100 and look at this piece.
But those are all getting moved around by the worms.
Right. And the worms in the water that move around. And
that's what you want. You want areas that are doing different things.
They're doing the work and they're being distributed through the whole system, right?
Yeah.
That's perfect. 40. There we are at 40.
That's the perfect amount of soil. I think that's a big difference in 3 versus everything else. This 3 is the biggest soil pocket. It's the closest to where the water initially comes in from the waterfall. So where the water initially comes in from the waterfall, it comes down and then the creek starts. And right when that first bend in that creek is where soil pocket number 3 is. It's the biggest pocket. And it bends on around.
Is there more perennials in that area?
Say what?
Is there more perennials in that area?
That's where the jasmine was, right?
I have that's where the jasmine is, the bush, like the jasmine,
there you go.
pink jasmine and the elephant ears. I have English ivy running down through there.
Oh, yeah.
So you've got more perennials in that area. That's good to attract more fungal connections.
Yeah, because they build more of those long-time relationships because they're going to live through more than one season.
Yeah. Oh, everything in there is perennial.
Yeah,
pretty much everything except for the cannabis.
Yeah,
bro, this organic matter is getting rocked, bro. Straight up.
Yeah,
it is getting rocked by saprophytic, by fucking microbes, by just the entire. This is a great learning piece, man. This is a good piece to show that the fungus is tearing it apart. You've got your little squigglies over here just decomposing, and you've got minerals when you hit it.
So when the fungal aspect, this is what I'm struggling with a little bit, the fungal aspect versus the bacteria aspect. The fungal is breaking down organic matter just like bacteria does, correct? Both of them. No.
So
they both
bacteria chews through the simple sugars, the quick, easy things. That's what bacteria choose through.
Okay, so the fungal is for the
more complex things.
Yeah, they all do though. They go after rock though when they need mineral, and they'll start tearing down sand, silt, and clay.
They all do it.
Yeah,
it's just the way they do it.
The key distinction to understand between fungi and bacteria is really predicated upon central intelligence. They have the ability to actually map out the gradient of the mineral composition of the substrate. They know where that piece of iron is when they need it. They map it out and understand where they're at in their own environment. And they actually feed the bacteria and farm and use them. So they're more of a steward of their own environment on a much larger scale than just the bacteria are. But they work together. And if you don't have fungi, then you're missing out because there's no brains to the bronze.
Right.
Yeah, exactly.
And
Exactly.
their C to N is also very, very different. So like the C to N on bacteria is what, 5 to 1. C to N on fungi can be 200, 300 to 1, depending on how old the hyphae are.
Is that a nematode there?
Yeah, it's like a bacterial nematode. Fat, happy, and just hanging out.
Yeah, even your nematodes are like in food comas, bro. They're just,
he's too tired.
Sleeping.
They got a food
Come
coma,
here,
bro.
hyphae pertinent here as long as my ear hair.
Get in on that mouth, though. I want to see that mouth. It's probably bacterial. It looks bacterial.
Oh yeah, yeah.
Like, what that mouth do though?
Oh, that's that.
Give me
Yeah,
a kiss. Yeah.
Yeah, that's definitely bacterial.
definitely bacterial. Yeah.
But like I say, your nematodes are even like, bro.
Yeah,
Do you
yeah.
think
You
Dune
know.
is actually about
and then look at
bacteria and
listen,
nematodes?
the saturation, bro, because nematodes
Yeah.
don't usually fluoresce like this.
No.
You don't normally see them fluorescing inside. And that's just a testimony to how the saturation and all the organic matter and microbes here, I guess, are somehow absorbing everything, and this nematode's eating it. He didn't get phased by that light. He's kind of like,
And that means by that nematode, these nematodes having all that color inside of them that's glowing, that means it's different types of minerals, different types of nutrients. And then once that comes out of that nematode, is that plant available?
yes, all day.
So that means that there's more micronutrients, more micro, everything all combined into one that's completely available. And the plant can then pick and choose what it wants,
when it wants, how much
it wants.
Yep. As long as it's there, right? And this guy's loaded with it. He's already shitted out. These little bacteria everywhere, by the way, they could be waiting. They could be waiting in the root zone for signals from the plant to say, wake up my calcium troops, right? My calcium troops will, the bacteria that go have the strongest enzymes to break calcium down will wake up, eat the sugar that the plant gives it, and go get calcium. And then this guy's got to eat them for the most part. They got to be eaten and pooped out, the poop loop, right?
Yep.
I think I got that from you. I heard you say that first.
The poop loop.
And that's the way it works. And so I mean,
so every single one of these little guys down here that's moving has a job. That each one of them is there for a reason. Each one of them is, like I said, they basically they're nonstop until they get eaten by something bigger than them, and then they get shit out, and then basically the cycle keeps. But I just have to maintain food for these guys and moisture for these guys to be able to do their job. So as long as I'm adding that sand and silt and clay as I see it being taken down, as long as I do that and maintain the flow of the water, this should continue to get better and better and better without me having to add like rock meat, like blood meal, fish meal.
I don't know if you can even be adding a whole lot of
things
really over time. I mean,
your plants are driving it mostly.
Your plants are going to feed the biology at this point, right? So your lights are feeding the plants, creating the sugars, creating the enzymes, and they're going to recycle, right? And as long as you're keeping your plant material minus what you're smoking back in that system, right?
That's really all I have to do then. So chop and drop.
Chop and drop. Exactly,
I just wanted to say.
So whatever buds you're pulling off of there, whatever fruit you're pulling out, that needs to be replaced over time. But you have a closed loop setup here that should just continue to do its job. Because you're going to add organic matter, crop or leaves drop or whatever,
Yeah, there's no reset.
There are currently
here.
30,000 described nematodes.
I'm sorry, what'd you say, sir?
There are currently 30,000 described nematodes.
Oh, wow.
They're estimated to be like millions.
Yeah.
Josh, if I wanted to blow this thing up, I would throw in blood meal. And then all of a sudden, everything will start acting crazy. Right? I throw the system off. If I start feeding this, if I took one of my ferments that you see me making with the fucking pomegranate that I dehydrate, I throw that in here. Things are going to be like, oh shit, we got extra food. We need to wake up and we need to get to work. And you'll see a scramble. And I can show you that if you want to see chaos, I can show you chaos in a completely unbalanced system that you'll be like, whoa, I don't want my shit moving like that.
And that would be bad, right?
Yeah, it would be bad.
Yeah, no, you're good.
This is great.
Like your job now, in my opinion, is just to maintain it, right? So do your chop and drop when you can, replant things. I would take if you're filtering out your water, get that filtered material back in here, right? So your fish poop, whatever you're filtering out, make sure it goes back into here to recycle all that.
Need some piles.
So just keep it closed loop. Again, all you gotta do is replace at that point whatever you're taking physically out of the room, right?
Yeah.
So if you're not taking stuff out of it, just recycle.
Checking it out.
Yeah.
I'm not taking anything out of it other than the bud itself. That's it.
And just keep it all in there. Keep your fish poop in there, right? I mean, filter it out and spread it somewhere on the top. Let it work its way down. Chop and drop your plants. If you want to slice them up or chip them or whatever. Fine.
Another reason this is so active like this is because I pull that string algae out of the creek and just put it in. I'm just placing it in
Again, you're now just a steward of the environment that you've created. And anything you do to upset that is going to upset that and not just let it do what it's doing, which it appears to do.
So if I did something like just something stupid and went throwing in something, and basically if I did that right now based on what y'all seeing, the system would just kind of overload itself and crash.
It would correct itself.
It would correct itself, correct. But I would basically crash it. Right, I'm in that sweet zone right now from what you guys are seeing.
Just steward it. Because
exactly.
you want to run out of clay, right? Your clay is not being removed and disappearing in the air. It's just getting moved around, right? You're fine. So as long as you're getting the organic matter back in there to refeed the fungus and the microbes over time,
we're good.
You're just maintaining that system, right? That is maintaining the system. That's your job now.
Yep.
If you go throwing stuff in there, you're just wasting your money, and you're just upsetting a balance that it's already created.
Yeah.
That's why. People keep struggling. You need this, you need that, and I was just like, no, I'm not doing anything.
I think you've done everything you need to do. I mean, I'm not saying anything.
Your job now is just to keep something living, growing in it all the time.
That living roots.
Yeah, the ones doing the work.
Well, and dead things on top. You want those dead things back on top.
It's dependent on the livestock load and the plant load too. So like you've got a buffer there, but you've got to maintain that same amount of like plant and animal activity as well. Yeah. Otherwise that could shift it out of balance.
Yeah, the top half could.
Yeah.
Like my arthropod. I can't say it.
Isopods and
all the bigger
tails and worms and
make sure they're happy and
everything
fed.
in there, bro.
But it's in balance at the moment. That's a great thing. Like most times you don't hit that.
and your worms eat this biology, by the way. They like nematodes, they like testate amoeba, they like all that stuff. They prefer that, right? So imagine what you're losing. You're not losing, they're balancing it.
Yeah,
it's all a balance, bro. You've got the balance there. You've got this thing.
Yeah, they're keeping everything
dialed in. You got it dialed in. In most instances, if I'm looking at compost, this is a whole different story. Oh, you need more. You're fungal. You're lacking on fungal. That's this not. This is a whole different beast. When I go.
Yeah, I think it really comes down to that moisture. It's really all about that water.
It's all about the water, bro.
Yeah.
Shit is everything,
it really does. If they have time and no dryback, and you just let them rip and roar, they balance themselves. You give them the food, you give them the home, the space, really enough space.
And you're including everything. Like you don't just have just plants and just microbes. You have plants and animals and microbes.
Everything.
So you have a complete system functioning as a system,
and that's what has to happen if we're going to get something like this, like what we're seeing, like what y'all have never seen before. That's how it has to be. We have to include the fish. We have to include all these different aspects, because when you bring it all together, we really start to see, the difference.
A couple of birds would be cool.
No. Yeah, exactly. If I could get a cage,
Get some guano drops going down.
Yes, yes. Like the whole food, the whole soil food web, all the way down, all the way up to the bird. To like I say, like to the lizard.
Man, look at all these rods.
That's unbalanced. That's unbalanced in chaos, bro. Right?
What is this?
This is me showing you what
unbalanced.
this is me showing you what an unbalanced system would fucking look like, right? It's just like,
holy shit.
You don't want this popping, dude. You know what I mean? And this is not even crazy.
So what are we looking at?
You're looking at one of the biofertilizers you see in my video. Now we want that. Okay. I want this as a biofertilizer to go into a compost. But my point of showing you this is that there's nothing stable here.
Yeah, this is something that would go into something that's like dead, like lifeless. Right. You
like sand.
Life to it. This is a life
like sand,
like pure sand, right? With no life to it. Yeah. Pure sand. Let's light it up, right? This is what I light things up with. But it's a whole different beast. There's no peace here.
You got a party, right? You got like a rave going on.
Yeah, this is
also got some phytohormones floating around too, right, Mo?
I've got
phyto. It also shows that there's excess nutrient that's still like being processed through.
Right, exactly.
Yours has found that place where it's like, well, we don't have this giant flush of nutrient always showing up to deal with.
Harmony.
Are all those nematodes?
No, those are all labs, bro.
Those are labs.
That's a flagellate.
Those are really long labs. They aren't usually
they're not your usual labs.
They're normally about a quarter-inch.
And then that's a flagellate there. Now, what I will say is amazing is that this has been on my desk for like a week and it's still not looking too anaerobic.
Not calmed down.
There's no crazy microbes that are usually known for being anaerobic.
Yeah.
Right? And then these guys all insisted. Remember, these guys were flying around the other day.
Mm-hmm.
They were all over the place.
Oh, look at that guy.
Oh, you know why? I put algae in here. Remember I told you?
Yeah, you dumped that algae in last night. Yeah.
That's right. So here's what happened last night. This was chaos. That's why I was expecting even more chaotic. But then I went and because it was going anaerobic on my desk, and this is what that looks like, right? So, and now you see the humic substance in that? You see how that's all brown?
Oh, yeah.
That's humic as shit. But it was going anaerobic, so I put purple non-sulfur and I put some chlorella in there to now start photosynthesizing and eating sunlight and putting oxygen into the system. And now these guys right here that were alive and well and zipping and fucking everything up, look at them. They're like
stable now, huh?
Right. And this is how I start figuring things out.
They went to sleep because the conditions aren't right for them to be active.
And that's the kind of testing I do where, that's what I love doing, right? I'll tell you what's happening when we go put, now I'm going to go put some of this stuff in your mic. When I'm done with testing your stuff, believe me, I'm going to culture them out because there are things in here that I want to add to my world, right? And hopefully whatever is going to survive is going to survive.
Mm-hmm.
And if it doesn't, it doesn't. But you see, I forgot that I did that last night because it was probably like 1 o'clock in the morning when I did it.
Yeah.
Hey, Landon came on here at like 10 o'clock laughing at us because we were still on here on impromptu call.
Gosh, just know that we're on Monday nights we rock like this, dude. If you ever want to dive
dude, I'm hooked.
y'all,
you're good, man.
You got the link, homie. So we're here doing.
Hey, so Josh, so too, in the chat I posted the link. So if you wanna, sometime tomorrow I'll upload this video to that link. So if you want to save that link, you can go back and watch this again.
Where are you going to put this?
It's in the main chat. There's a little chat button on the bottom, and I'll pull up a window.
Yeah, it's a Google. I'll email it.
Email it
to
you. Man.
Yeah.
Okay.
Appreciate it.
We're every Monday night, so you can always just use it. It's the same link.
Yeah, okay. I got you in the chat. Right.
So now what I'll do is I'm going to go back to the first sample since it's been sitting in water for some time. This is what I do, and it's like, let's see what happens since it's been in water for 34 or an hour and
for an hour.
Damn, 9, 10, 11. Bro, we've been on for 2.5 hours. So let's see what happens after 2.5 hours, right? Let's see what happens after 10 hours, and then once it gets stupid, let's see what happens if I put algae in it or purple non-sulfur bacteria, labs, right? That's all part of it. And by the way, Josh Kennedy here. He was he's the aquarium, he's dealt with aquariums as well. So Josh, before you jumped on, he was talking about how he started. He wished he knew about labs when he had the aquatic world and he was doing
I agree with that 100%.
Yeah, it's been a game changer for me. if used properly, you don't need much. But I mean, it's
what was the biggest change that you noticed when you started using the labs?
I noticed the fish and the turtle shell. The turtle shell and the fish scales on everything were just super bright.
That's cool.
Everything super healthy. The water clarity cleared up tremendously. The grasses were greener. Just everything just seemed to go back into normal. Like I was getting this fungal like brown spot on my
Giant Toms.
on my elephant ears. Yeah. And I just take a shot glass and just boosh right around the base of the elephant ear.
Yeah.
It went away. Like it just like took care of it. And that's all it took. And then I just douse it in the pond, douse it in the creek like once a month, just a shot glass, just a little shot glass. I make fresh labs here, right here with the rice and milk, boosh right into the water, and it's been a game changer.
Yeah, I remember first hearing about it from Stephen Reisner. And he was saying he ran labs in his aquaponics, and he's like, man, the fish nip at each other. We don't get ick. We don't get protozoa infections. Like, it's game changer.
I heard the same interview. I heard the same. I've talked with Stephen quite a few times. He's had me on his podcast, and yeah, he's a pretty cool dude, and he knows a lot of stuff about that aquaponics stuff, man. He's done some research.
He rattles off some crazy science sometimes.
Yeah, like he loses me quite a bit, but I pick up on a few things. The more I listen to it, the more I pick up. And the lab was one of the things that was just a game changer for me. I was like, holy shit, that's like he was using his aquaponics. I was like, wait, what? You do what? And I was like, oh shit. And then I did it in my aquarium, and I was like, oh shit. And then I did it out there in the pond, and I was like, yeah.
And on the plants.
Yeah, I was like, I'm putting that shit on everything now.
We're back to area 1.
Yeah, notice the nematodes have all calmed down. When we were here before, they were all really active. They've all really settled down. And is that one that's now?
That's an egg, right? Is that an egg?
Yeah.
That's an
egg. Nematode?
I don't know.
I don't know.
It looks segmented big time.
Yeah, it doesn't look like
unless it's like in an ultra early stage of nematoism. No, I'm kidding.
Is this a different? Is this the same slide?
This is no. I went back into the tube.
Okay.
And then pulled another sample.
So you just see something different every time. We'll see.
Well, the longer it sits here, the more chaos is going to become of it.
Okay.
Because there's going to be we start losing oxygen, right? Yeah, it's only been like 10 hours, but by tomorrow I'll look at it again and be like, all right, it took 15, 20, 30 hours for the system to start bugging out. I've seen stuff hold for days where it's like, yeah, we've got so much oxygen in the organic matter that it takes quite a while for everything to officially start going anaerobic.
Nuts.
Really well, remember, it's not just how much oxygen may be in the OM, but also the antioxidant and the ORP value of the water itself.
What is that, Mo?
That's gonna be algae, bro. Some type of moss.
If you epi, that's gonna be like a bit of red.
That's why Pinto's recipes are calling for grape juice.
Is that algae that's senescing, Mo? It's turning because it's kind of a purpley color.
It could be senescing. Yeah.
Why grape juice?
The more antioxidant value and particulate, molecular value per droplet of volume.
Okay.